Hematoxylin and eosin stain is one of the principal stains in histology. It is the most widely used stain in medical diagnosis and is often the gold standard. For example, when a pathologist looks at a biopsy of a suspected cancer, the histological section is likely to be stained with H&E and termed H&E section, H+E section, or HE section. The staining method involves application of Hemalum, a complex formed from aluminum ions and hematein. Hemalum colors nuclei of cells blue. The nuclear staining is followed by counterstaining with an aqueous or alcoholic solution of eosin Y, which colors other, eosinophilic structures in various shades of red, pink and orange. A mnemonic often used to remember the coloring process is acidic attaches Acidophilic applying Auburn and basic binds Basophilic becoming blue. The staining of nuclei by Hemalum is ordinarily due to binding of the dimetal complex to DNA, but nuclear staining can be obtained after extraction of DNA from tissue sections. The mechanism is different from that of nuclear staining by basic dyes such as thionine or toluidine blue. Staining by basic dyes occurs only from solutions that are less acidic than Hemalum, and it is prevented by prior chemical or enzymatic extraction of nucleic acids. There is evidence to indicate that coordinate bonds, similar to those that hold aluminum and hematein together, bind the Hemalum complex to DNA and to carboxy groups of proteins in the nuclear chromatin. The eosinophilic structures are generally composed of intracellular or extracellular protein. The Lewy bodies and Mallory bodies are examples of eosinophilic structures. Most of the cytoplasm is eosinophilic. Red blood cells are stained intensely red. The structures do not have to be acidic or basic to be called basophilic and eosinophilic. The terminology is based on the affinity of cellular components for the dyes. Other colors, for example yellow and brown, can be present in the sample. They are caused by intrinsic pigments, for example melanin. Some structures do not stain well. Basal laminae need to be stained by PAS stain or some silver stains, if they have to be well visible. Reticular fibers also require silver stain. Hydrophobic structures also tend to remain clear. These are usually rich in fats, for example adipocytes, myelin around neuron axons, and Golgi apparatus membranes. See also, Papanicolaou stain, other popular staining technique, cytopathology, eosin, acid-fast, references, Godwin A. Bureau. Histochemical uses of hematoxylin, a review. JPCS 124-34. PDF Baker Jr. Experiments on the action of mordants. 2. Aluminium hematein. Quart. J. Microsk. Sci 103, 493-517. Kyan and J. A. Histological and Histochemical Methods, Theory and Practice. 4th ed. Bloxham, UK, Scion. Lily Road, Pozzolito P., Donaldson Point nuclear stains with soluble metachrome mordant lake dyes. The effect of chemical end-group blocking reactions and the artificial introduction of acid groups into tissues. Histochemistry 49, 23-35. Llewellyn B. D. Nuclear staining with alum hematoxylin. Biotech. Histochem. 84, 159-177. Marshall P. N., Robin R. W. The Mechanism of Action of Mordant Des, a study using preformed metal complexes. Histochemy 35, 361-371. Putler H., Mello and S. N., Waldrop F. S. Application of current chemical concepts to metal hematein and brazilene stains. Histochemistry 85, 353-364. External links, Sigma Aldrich H&E Informational Primer, Protocol, Routine Mayer's Hematoxylin and Eosin Stain, Hematoxylin and Eosin Staining Protocol, Rosen Lab, Department of Molecular and Cellular Biology, Baylor College of Medicine, Step-by-Step Protocol.